Okay, so welcome to this next video in the playlist on synaptic mechanisms. In this video, we're going to continue our discussion of endocytosis, and we're going to discuss adapter protein complexes in this video. So we're going to have a discussion of adapter protein complexes. So we'll begin with just a brief summary of what we've discussed in previous videos about endocytic motifs, and then we'll discuss adapter protein complexes in more detail. Okay, so let's begin then. So, if we want to endocytose a protein from the plasma membrane, okay, so let's say we have our phospholipid bilayer here, and we have some protein in the plasma membrane that we want to endocytose, okay, so let's say this is the extracellular fluid here, and this is the cytoplasmic side here. Right, so we've discussed that in order to get endocytosis, this protein has to have exposed on its cytoplasmic domain a certain sequence of amino acids which tells the cell that this protein, this target protein here, so let's say this is our target protein, that tells the cell that this target protein needs to be endocytosed. Now these sequence of amino acids which tell the cell that this target protein needs to be endocytosed, they are called endocytic motifs. Okay, so let's put that down here. So let's say somewhere in this intracellular domain here will be an endocytic motif. Okay, and there are two endocytic motifs that we looked at in the previous video. So one is that you would firstly have the amino acid asparagine, so I'll use the single letter amino acid codes. So N is the single letter amino acid code for asparagine. So this is asparagine, which is basically just the primary amide of aspartic acid. Then you have anything, followed by anything, so we just put X there. And then finally Y, which denotes the amino acid tyrosine. So this is an example of an endocytic motif that is very common. This is the one that was in the LDL receptor cytoplasmic domain, and is the one where you can get this tyrosine residue mutated to a cysteine residue, and that produces familial hypercholesterolemia. Okay, so the other endocytic motif that we saw was you had a tyrosine in position 1, followed by anything, and then Phi, which is not a single letter amino acid code, but instead means that you just need a bulky hydrophobic amino acid. And examples of bulky hydrophobic amino acids are the amino acid phenylalanine, which is given the single letter amino acid code F, so this is phenylalanine, or another example is isoleucine, I. Okay, so both of those sequences, whether it's this one here or this one here, they are endocytic motifs. So they are combinations of amino acids within this polypeptide that are exposed, and that's important, they need to be exposed on the cytoplasmic aspect of the protein, basically. So they need to be exposed to the cytoplasm. Then what's going to happen is this protein is going to be endocytosed. How is the question? Well, basically, there are protein complexes which recognize these endocytic motifs here. So let me draw arrows to these endocytic motifs here, which recognize these endocytic motifs and will bind to them, and then when that adapter protein complex binds to that endocytic motif, it will then recruit other proteins which are involved in the endocytosis of this protein. So, now let's study the properties of these adapter protein complexes. So, there are different types of adapter protein complexes, and they're involved at different membranes. Now, the main two types are the adapter protein complex, denoted AP, of the first type, AP1, Then there's also the adaptor protein complex of the second type, AP2, and then another important one is the adaptor protein complex 180, and I don't know quite what happened there. I assume 180 has something to do with its molecular weight. I don't think, they went through in chronological order. I don't think there's quite 180 of these things. So I think the 180 there is to do with the number of kilodaltons that it is, rather than what order it was discovered in. Now, they all act at slightly different membranes. So the one which is important at the plasma membrane is this adaptor protein complex 2. So that's the one we're going to study, because we are looking at endocytosis that is occurring at the plasma membrane of our neuron, for instance. Okay, so let's study adaptor protein complex 2. Often denoted just as AP2. Right, so adaptor protein complex 2, as the name suggests, it is a protein complex, so it is not just made up of a single protein, it is made up of multiple proteins that have joined together to form a protein complex. Now, let's have a look at the structure of this protein complex. So, if I draw this out for you. So, it has this sort of Mickey Mouse appearance, is what it's often compared to. And you'll see how that's actually relevant for what it's called later. It's not quite called Mickey Mouse, it's not quite Sonic Hedgehog. Well, I didn't have the same people naming it as Sonic Hedgehog had naming it. So it is an adaptor protein complex, but these structures here are actually known as the ears often. Okay, right. So let's draw it here. So there are multiple different ways of drawing it, but this is my favourite way of drawing it. Okay, so here we have these two, and it's not perfectly symmetric, but never mind, it should be. Okay, so this is the alpha subunit, this is the beta subunit, then you have a subunit sitting in the middle here, known as the mu subunit, so mu subunit here, and you have another subunit sitting in here, known as the sigma subunit, and this overall is the adaptor protein complex. Right, so each one of these subunits, the alpha, the beta, the mu, and the sigma, they are all adaptin proteins. So this is the alpha adaptin protein, this is the beta adaptin protein, this is the mu adaptin protein, and this is the gamma adaptin protein, and they assemble together to make this adaptor protein complex. So let's colour them all in different colours. So here in purple is the alpha adaptin protein. Okay, whoops. And like that. Then in the middle here we have these mu adaptin protein here, which is in blue. Okay, and we also have the sigma adaptor protein in green here. And then finally in orange we have the beta adaptin protein. And together they make up the adaptor protein complex 2, often denoted as the AP2 complex. And this is what is going to recognize the endocytic motifs on the cytoplasmic domain of these proteins that we want to endocytose. Okay, now some more important things is we can see that there are two different distinct portions of this protein. So the portion down here, okay, all of this portion here. So let me, well in fact, more than just this, we need to also include, in fact, all the way up to here as well. So anything that's not these two ears coming off. So let me highlight this. All of this bit, shown in red here, ignore that line that I've managed to draw through the middle of it. All of this bit here is what's known as the core domain of the adaptor protein complex. So this is the core domain of the adaptor protein complex 2. So core domain of AP2. And by the way, the adaptor protein 1 and the adaptor protein 180, they have almost identical structures. Okay, so although we're studying this for the core domain AP2, although we're studying it for the adaptor protein complex 2, the adaptor protein complex 1 and adaptor protein complex 180 have very similar structures. Okay, right. Now these bits sticking off the edge, which I called the ears, that is what they're referred to as. They are called the ears of the protein complex. Okay, or more properly, they're called the appendages. So equals the appendages of the AP2 complex. Appendages. Okay, so you have the core domain and then you have these two appendages, the alpha appendage and the beta appendage, or the alpha ear and the beta ear. Right, okay, now, what does this protein complex do? Well, we know what it needs to do. It needs to bind to the endocytic motif. Now, it is the core domain of the AP2 complex which binds to the endocytic motif. Okay, so the core domain here is going to bind to the endocytic motif, and the ears, the appendages, are going to interact with other proteins that are then going to come in and cause the target protein to be endocytosed. Right. However, before we can actually get it binding to the endocytic motifs, we need to get it to the membrane. So how do you target the adaptor protein complex to the membrane? Well, the answer to that question we'll explore in the next video.